Hello again, everyone. Today, we want to explore one of the more difficult concepts in physiology. We want to begin looking at electrical properties of cells. And in particular, we want to look at something called the resting membrane potential. Now, this is a topic that many students find challenging. So as we go through this session, I'm going to go very slowly, but I'm going to encourage you to do your reading and to do the different calculations and the different things that I ask you to do. And also to look at other videos and really try to understand what is taking place so that you can get a good grasp of how resting membrane potentials come about. Now, let's just start by looking at this little diagram on the left. In the diagram, you can see a neuron, and then we've highlighted a very small part of the neuron to look at it in a little bit more detail. And we're using a voltmeter. Now, a voltmeter is something that measures potential difference or the separation of charge. And we use a very small microelectrode. We put one electrode on the inside of the cell and one electrode on the outside of the cell. And what you immediately notice when you do that is the inside of the cell is relatively negative with respect to the outside of the cell. In other words, there is a separation of charge. Now, the resting membrane potential by definition is then the potential difference across the cell membrane when it is at rest or when it's in a non-excitable state. All cells in the body have a resting membrane potential, but not all cells are excitable. Only cells that can generate action potentials are considered excitable. An example of this would be a nerve cell or a muscle cell. And what we're going to do for the rest of this lecture is look at the different things that are responsible for the generation of this resting membrane potential. Now, the first thing we need to understand to appreciate the resting membrane potential is it's really an electrical system that is being set up here by the body. It's a biological system, but it's also an electrical system. And you can look at the resting membrane potential simply in terms of electrical currents. We're not going to do that today, but I just want you to appreciate that the cell membrane, which you've studied extensively in the past, and which is a bilipid layer, the presence of that impermeable bilipid layer causes it to function like a capacitor because that layer cannot conduct electricity. So it functions like a capacitor and it's able to store charge on either side. And that's how you end up with this membrane potential. That membrane potential can actually be given by the electrical equation V, voltage, is equal to the charge over the capacitance of the membrane. But like I said, we're not going to spend too much time upon that. But I just want you to appreciate the electrical properties that are being built into the cell membrane and how these can then lead to the resting membrane potential. So here's a couple of questions. What is the normal range for the resting membrane potential? All biological systems exist in a normal range. And for the RMP, it lies between -60 and -90 millivolts. And different cells tend to have particular ranges. On average, we say that the typical neuronal cell has a resting membrane potential of about -70 millivolts. But some cells, like red blood cells, have been known to have values as high as -10 millivolts. And then other cells, not so much in the human body, but certain bacteria or certain plant cells, they can have an RMP as low as -200 millivolts. But for the majority of cells in the body and the ones you will be studying, those will be muscle cells and neuronal cells. We can say the normal range is -60 to -90 millivolts. Why is the value negative? Well, this is a convention and it reflects the fact that the inside of the cell is negative. And in fact, it affects the fact that when we measure it, by definition, we'd say it's the voltage on the inside of the cell minus the voltage on the outside of the cell. Now, if we're going to understand what causes the resting membrane potential, I just want to talk to you about the different types of ion channels that exist within the cell membrane. Now, you remember the structure of the cell membrane very well, the phospholipid bilayer. And we know that phospholipid bilayer is impermeable to charged particles. But we also know charged particles and ions are essential for bodily function. And so the body has a system of channels, integral proteins, that span the entire cell membrane and allow certain ions to pass through and therefore enter and leave the cell. Now you can think of these ion channels like gates, and you can actually think of the gate to your home or your house if you have a gate to your home or your house. And it's going to help you to understand different types of ion channels that exist. So let's begin looking at the 4 types of ion channels. We want to start here in the bottom left-hand side. Here you'll notice an ion channel that is always open. Sometimes these are referred to as leaky channels or passive channels. Now this is like the gate to your house, and the gate is always open. No one ever closes it. And the result is ions can move freely in and out along those channels. Now let me just say that there's one thing to remember about these channels. Even though they're always open, like other channels, these channels are specific. That means you have leaky channels that only allow potassium to pass, or you have leaky channels that only allow sodium to pass. And therefore, even though they're always open, they don't let all ions pass through. There are specific channels for potassium, for sodium, for chloride, and for other substances. So that's the first type of channel that we want to talk about, the leaky channels. Well, the second channel we want to talk about, let's look at the bottom right. That's our voltage gated channel. Now you can think about this like your gate, and some of you have gates at your home that you control with electricity. There's a button that you press and they're always open. They're electric gates. Well, the voltage gated is something like this. It responds to changes in charge. These gates will open when there's a depolarization of the cell membrane. When the membrane potential actually changes, it causes a configuration change in the channel, and the channel opens. And a very good example that we're going to study later on is the voltage gated sodium channel, and that's what you're seeing here. So normally this voltage gated sodium channel will be closed to sodium. But when there's a membrane depolarization, when the resting membrane potential moves from -70 millivolts to say -40 millivolts, this causes the channel to open and it allows sodium to flow through. Note that this channel is also specific, but now it's not always open, but it's dependent upon a change in voltage. Let's look at the 2 channels at the top of our diagram. To the left, we have the ligand-gated channel. Now, these channels are also specific, but in order for them to open, a chemical must bind. And a good example of that would be a neurotransmitter like serotonin or dopamine or acetylcholine. When the neurotransmitter binds, as you can see in this diagram, that causes the channel to open, and that now allows the ions to flow in and out through the channel. The last channel is much less common and only found in certain specific parts of the body, often in the skin, and that's the mechanically gated channel. And these channels open when there's some type of deformity that takes place in the cell membrane, primarily as a result of direct pressure. And this will then cause a conformational change in the channel, and it will open and allow ions to flow through. Again, note that these channels, like all the others, will be specific. And so those 4 channels are going to be very important for us to understand a lot of the physiology that you'll be looking at during your course. In particular, as we look at the resting membrane potential, we're going to focus a lot on the leaky channels. And we're going to see their importance in understanding the resting membrane potential. So the resting membrane potential, we've already said for most nerves, it's approximately minus 70 millivolts. So we want to find out how is it achieved and how is it maintained. And the key principle that we're going to be focusing on is something called electrochemical equilibrium. Electrochemical equilibrium. We know in biology, systems always want to move to a state of equilibrium. Now, you've already studied and you already know that the number of ions or the concentration of ions inside of the cell is different from the concentration of ions on the outside of the cell. And you also know that once there's a concentration difference, ions will always seek to move down their chemical gradients. And we want to talk about how that uneven distribution of ions ends up being responsible for the generation of something called the electrochemical equilibrium. To do that, we're going to look at a very specific example of something called a concentration cell. And I want you to imagine that this is an actual cell. We can consider side 1 to be the inside of the cell, and we can consider side 2 to be the outside of the cell. And for the purposes of this particular explanation, we want to say that this membrane that separates side 1 and side 2, let's say that it's permeable, but it's permeable only to potassium. Okay, only to potassium. Now you note that potassium has 100 millimoles or milliequivalents, whichever one you want to use, on side 1. And on side 2, there are 10 milliequivalents. Now I want to ask you, what will happen to the potassium? And I also want you to think about, when equilibrium is achieved, what will be the concentration of potassium on side 1, and what will be the concentration of potassium on side 2? You can pause the video and think about that for a little bit and actually write down what do you think the concentrations will be. Now, according to diffusion, substances are going to move from an area of high concentration to an area of low concentration. So we would expect the potassium to move from side 1 to side 2. That makes a lot of sense. And if nothing was involved, if there was no change, no other situation that we had to consider, we could say that at the equilibrium point, you should have approximately 55 millimoles of potassium on one side and 55 millimoles of potassium on the other side. Now, that's what we would expect if the only force acting upon the potassium was the diffusion forces or the chemical forces. But potassium has something very unique about it, and it's common to many other substances. That's it's an ion, and it has a certain charge upon it. And we know charge, whenever you talk about the term charge, you're talking about electricity. So what's going to happen when we start to think about charge? Well, potassium is positively charged. And when 1 millimole of potassium moves from side 1 to side 2, you end up with 99 millimoles on side 1 and 11 millimoles on side 2. Now, I don't want you to think about chemical gradients now. I want you to think about electrical gradients. As the concentration of potassium builds up on side 2, that's an increase in positive charge. And we know like charges repel each other. So as potassium builds up on side 2, there's another force that comes into play, and that's an electrical force. And the electrical force is actually pushing the potassium from side 2 to side 1, because the positively charged potassium over here is repelling the positive charged potassium here that's trying to come into side 2. So what we understand from this is there are actually now two forces acting on the potassium. There's a chemical force of the concentration gradient moving the potassium from side 1 to side 2. But there's also an electrical force that's opposing that chemical force, and it's pushing the potassium in the opposite direction. The fact that potassium is charged means we cannot simply think about the chemical gradient. We also now have to think about this electrical force. Now there's going to come a point when the chemical force is actually balanced by the electrical force. And that point usually arrives long before 55 millimoles on each side. And at that point, we say the system is in electrochemical equilibrium. Electrochemical equilibrium. It's not simply a chemical equilibrium due to diffusion, but it's an electrical equilibrium and a chemical equilibrium combined. The two forces have now come into equilibrium, and there's no net movement of potassium. And what you'll note in this situation is, as a result of that force, there's an uneven distribution of the ions in terms of their charge. And so one side of the system will have more charge than the other. And this is exactly what happens to set up the resting membrane potential. Now, students tend to find this a difficult concept, so I'm just going to go through it again fairly quickly to help you understand. So at time 0, we have 100 millimoles of potassium on one side, and we have 10 millimoles of potassium on side 2. Potassium will therefore flow down its diffusion or chemical gradient from side 1 to side 2. However, every time a positive charge moves from side 1 to side 2, it causes more positive charges to accumulate here on side 2. And the end result is the buildup of an electrical force moving in the opposite direction that opposes the chemical force. The point at which these two forces are equal are called the electrochemical equilibrium. But when that equilibrium occurs, there's an uneven distribution of ions, and therefore that's what's responsible for that separation or difference of charge that sets up the resting membrane potential. So you can see in the diagram there on the left that there's a chemical driving force that's pushing the potassium out of the cell, but there's an electrical driving force that's pushing the potassium into the cell. And the point where they are balanced, we call that the equilibrium potential, or sometimes we call it the reversal potential, because if the electrical driving force exceeds the chemical force, it will cause a change in the direction of the movement of potassium. You can actually measure that force with instruments, but people have studied the things that are responsible for creating that force. And there's an equation that allows us to generate what is the electrical potential that opposes a particular chemical force, or what electrical force will be needed to balance a particular chemical gradient. The name of that equation is the Nernst equation, as you can see on the slide. And that equation is given by this formula. The equilibrium potential for any ion is equal to minus RT over ZF by the natural logarithm of the ionic concentration on the inside versus the ionic concentration on the outside. And those letters there should be fairly familiar to you. R is the universal gas constant. T is the absolute temperature measured in degrees Kelvin. Z is the valence of the ion. Valence refers to the charge, and not just the size of the charge, that is +1, but also the particular polarity of the charge. So potassium, if I were to ask you, what is the valence of potassium? You should tell me +1. If I should ask you, what is the valence of chloride? You should tell me -1. If I ask you, what is the valence of calcium? You should tell me +2. And finally, F is a Faraday constant. Now, this seems like a fairly complex equation, but often in biology, we can simplify it because certain things are common. And so for biological membranes at a temperature of approximately 37 degrees, that equation simplifies to the following. The balancing electrical force, the balancing voltage for a particular ion is equal to minus 61 millivolts over the valence by the log of the concentration on the inside over the concentration on the outside. Now that's a simplified form of the equation, but I want you to keep that in mind because it allows you to make very simple calculations. And what I want you to do now is I want you to pause this video and I want you to look at the concentration of potassium on the inside of the cell and the concentration of potassium on the outside of the cell. And I want you to put those values into this equation and therefore calculate the equilibrium potential for a cell that is permeable only to potassium. So pause the video, plug in the values for potassium into that equation and calculate the equilibrium potential for potassium. What you're calculating there is the actual electrical force that is needed to balance the chemical force working on potassium. Now, if you did that, you should have had some sort of calculations like the ones that you're seeing before you now. In this example, we use the concentration of potassium on the outside to be 4, and we use the concentration on the inside to be 140. And when you go through the calculations, what you should have found was that the balance equilibrium or the equilibrium potential for potassium was approximately minus 94 millivolts. So what we're saying is that if a cell were only permeable to potassium, because of that balance, that electrochemical equilibrium, a separation of charge would be set up, a force would be set up of minus 94 millivolts that would balance that chemical gradient. And therefore, when you use your voltmeter to measure the resting membrane potential for that cell, what you'd get is a value of minus 94. And so we can see that the uneven distribution of ions and the fact that they're flowing down their chemical gradients is able to set up this electrochemical balance. Now, what I'd like you to do is imagine a cell is only permeable to sodium. And I want you to use the Nernst equation now to calculate the equilibrium potential for sodium. And then I want you to do the same thing for chloride. And the reason we're doing this is for you to understand the principle and also to get accustomed to using these equations. Now, this equation that you see on your screen looks very, very complex. But the principle behind this equation is actually quite simple. In the examples we were using previously, we imagined that the cell was only permeable to one ion. In the first example that we focused on, we imagined that the cell was only permeable to potassium. And then I said, well, focus on sodium and chloride. But in reality, the cell is permeable to many ions, many ions. And in order to calculate the resting membrane potential, we have to use the weighted average of the equilibrium potential for all of the ions. And that is given by this equation called the Goldman-Hodgkin-Katz equation. And what you can see basically is that it's very similar to the Nernst equation, but it considers the permeability of each ion alongside its equilibrium potential. So here you have the permeability of potassium, the permeability of sodium, the permeability of chloride. And what you can then see from this particular equation is the ions that have the largest permeability are the ions that are going to have the greatest effect on the resting membrane potential. When you actually do experiments, you find that the Goldman-Hodgkin-Katz equation is the best equation for modeling real-world values. So even though the Nernst equation can tell us the equilibrium potential for one ion, in order to get the resting membrane potential for a cell, it's better to utilize the Goldman-Hodgkin-Katz equation. And the equation you see at the bottom is a simplified form of the Goldman-Hodgkin-Katz, at approximately 30-something degrees, focusing just on potassium and sodium. Now remember, I told you that you can also consider the cell from an electrical point of view. And if you're thinking about the cell from an electrical point of view, you won't think and talk about permeabilities, but you'll talk about the term conductance. Conductance is the inverse of resistance. It's how easily will current flow through the cell membrane. And so you can talk about sodium conductance, and you can talk about potassium conductance. And if you use conductance, you can come up with a formula for the resting membrane potential that's based upon the different conductance values for each ion. And this equation is called the chord conductance equation. And it states that the resting membrane potential is equal to the conductance of sodium multiplied by the equilibrium potential for sodium, plus the conductance of potassium multiplied by the equilibrium potential of potassium, plus the conductance of chloride multiplied by the equilibrium potential for chloride, divided by total conductance. And we're going to be doing some examples of how we can utilize this question, this equation, in another session. So I want you just to do a little bit of reading and understand the chord conductance equation. It's based upon the same principles as the Goldman-Hodgkin-Katz equation, but now it's using electrical systems and the flow of ions, which is actually an electrical current, to calculate the resting membrane potential. Now, at rest, the permeability of the cell membrane is much greater for potassium than it is for sodium. And the reason for this is there are a lot more potassium leaky channels than there are sodium leaky channels. And different studies have come up with different values, some as low as 50 times more potassium channels, some as high as 400 times more potassium channels. But the basic principle is that there are a lot more potassium leaky channels than sodium. That means the cell membrane is a lot more permeable to potassium than it is to sodium. And you can see that on this diagram here. As the ion concentration increases, the flow through the cell membrane for potassium is a lot greater than it is for sodium. As the chemical gradient increases, a lot more potassium is able to move through the membrane because there are a lot more ion channels. You can think of these ion channels a little bit like the facilitator proteins that we spoke about in facilitated diffusion. And because there are a lot more for potassium, you can have a lot more potassium moving in. What that means is that the resting membrane potential is more influenced by the equilibrium potential for potassium than it is for sodium. So when you calculated the equilibrium potential for potassium, you would have found it to be around -90. When you calculated it for sodium, you would have found it to be about +60. But when you actually look at the measured value, it tends to be around -70. It's a lot closer to the value for potassium because potassium is a lot more permeable through the cell membrane than it is for sodium. So its weighted effect upon the RMP is a lot greater than the effect of sodium. When we study action potentials, we'll actually see that this changes, and this has enormous consequences for the cells. But when the cell is at rest, you can say that potassium has the largest effect upon the resting membrane potential. Now, this actually has certain implications for intravenous therapy. Because you know, when you add intravenous fluids into the body, you can change the solute concentration of the extracellular fluid. And what I want you to do now is I just want you to do a little exercise. Go back to the example where you calculated the Nernst equilibrium potential for potassium. But now I want you to change the potassium concentration in the ECF. I want you to double it, and I want you to calculate the equilibrium potential. When you do that, what you should realize is the equilibrium potential now gets a lot less negative. It will move from -90 to something a lot less negative. And this actually makes the cells a lot more excitable because they're closer to their threshold for firing. And you'll understand more of that when we do action potentials. I want you to do another little study. I want you to double the permeability of the cell to sodium. Double the permeability of cell to sodium. And I want you to think, this is now like a thought experiment because you're not going to be able to use actual figures. But I want you to think, what effect would that have on the resting membrane potential? If you made the cell double, twice as permeable to sodium, would the resting membrane potential stay the same, or would it actually change? And here you see an article from a newspaper. This is many years ago, over 20 years ago. And it describes a fact that by accident, somebody was given potassium chloride in their intravenous fluid instead of sodium chloride. The end result was it caused death because there was a cardiac arrhythmia. The cardiac cells started to fire in a strange manner. Based on what I've told you, I want you to be able to explain what happened in this particular case. So you can read through it and you can think about how introducing higher levels of potassium chloride into the extracellular fluid might affect the heart cells, which are excitable cells. Okay, so we've spoken about the fact that the resting membrane potential is a result of the uneven distribution of ions and their movement down their concentration gradients. And that's a primary factor that's responsible for the resting membrane potential. But there are two other factors that I want to just mention that do contribute something to the resting membrane potential. The first other factor is the sodium-potassium ATPase pump. Now that pump, as you know, is responsible for helping to maintain ionic gradients across the cell membrane. And that pump, as you know, it's able to pump, as you can see in the diagram, 3 sodium ions out of the cell and 2 potassium ions into the cell. It's an ATPase because it's moving sodium against its concentration gradient and it's moving potassium against its concentration gradient. So the sodium is going from a low concentration on the inside to a high concentration. And the same thing for the potassium, just moving in the other direction. But I want you to note that this pump actually moves 3 sodium, but it only brings 2 potassium to the inside of the cell. Now, if you think about that in terms of charge and not in terms of the actual ions themselves, for every cycle of that pump, it's pumping 3 positive charges out and it's only bringing 2 positive charges in. So we can say that the pump itself is electrogenic in nature. The pump itself is actually causing a separation of charge. And for every cycle of the pump, it's actually putting 3 positive out and only bringing 2 in, meaning that it's creating a net negative of 1 on the inside of the cell. So the actual effect of the pump, the primary role of that pump is to maintain the balance of the distribution of ions. But in doing so, it actually helps to set up the resting membrane potential. And it increases the RMP by approximately -5 to -10 millivolts. Some values will tell you as low as -4 millivolts. And we call this the electrogenic nature of the pump. So the pump has a crucial role in setting up the RMP because it helps maintain the distribution of ions, but also has a small contribution to the actual negativity of the value on the inside. The final factor is something called the Gibbs-Donnan equilibrium. Now, inside of the cell, there are large intracellular proteins. These proteins are negatively charged, but they cannot cross the cell membrane The membrane is impermeable to them. Now, the presence of these proteins allows the inside of the cell to remain electrical neutral without having a high concentration of anions. So you find the chloride concentration will be fairly low. But what the Gibbs-Donnan equilibrium says is the product of diffusible ions on one side of the membrane must be equal to the product of diffusible ions on the other side of the membrane. That's a law that's been discovered. Now, because of this, and because the large proteins cannot move, it sets up an unusual occurrence, as you can see in this diagram. So here, we have sodium chloride in the extracellular space, 150 millimoles. But in the intracellular space, we have 150 millimoles of sodium. But that's balanced not by chloride, but by these largely negative proteins. Now, the end result, the proteins can't move, but the sodium and chloride can move to a certain extent. Chloride will move down its concentration gradient, and sodium will follow it. And eventually, the balance point will be reached where the products have to be the same on either side. So here it's reached with 100 millimoles on either side. But now it's balanced by 200 millimoles of sodium on this side and 50 millimoles of chloride. And the electrical neutrality is achieved by the 150 millimoles of largely those negative ions. What this then creates is a slightly more negative charge on the inside of the cell. Now, at this point, the cell isn't in total balance because the osmolarity in the inside of the cell is actually increased because of the number of particles. And there would be a movement of water into the cell to cause it to flow, to swell. However, in real life, the Gibbs-Donnan equilibrium is not the only thing acting upon the cell. We've just seen that there are other forces due to the electrochemical equilibria acting on the cell. And so you never have this osmotic imbalance and you don't have the cells swelling. However, the presence of these proteins and the result of the Gibbs-Donnan equilibrium means that it contributes approximately minus 10 millivolts towards the RMP. The presence of the negatively charged proteins on the inside of the cell that cannot move contribute approximately minus 10 millivolts towards the RMP. So you can think about it like this. If the RMP of a nerve is approximately minus 70 millivolts or minus 75 millivolts, about 5 millivolts is due to electrogenic nature of the sodium-potassium ATPase pump. About 10 millivolts is due to the negatively charged proteins and the Gibbs-Donnan equilibrium. And the rest will due to the balance of the ions flowing down their electrochemical gradients, of which potassium has the greatest effect. Now, the Gibbs-Donnan equilibrium is also a little complex, so I'm going to ask you to read and understand it. So those are the three factors that are responsible for the resting membrane potential. You have the uneven distribution of ions and their movement down their electrochemical gradients and the balance points. And studying that, you need to know the equations, the Nernst equation, the Goldman, hodgkin, Katz equation, and the quad conductance equation. And you need to understand how to do simple calculations. And then you have the other two factors, which are much smaller, which were also important to understand. The electrogenic nature of the sodium-potassium ATPase pump, and also the contribution of the large negatively charged proteins inside of the cell, and the effects of the Gibbs-Donnan equilibrium. As I said at the beginning, this can be a difficult topic. And what I'd like you to do is I'd like you to listen to this video again, look at other videos that you can find on the internet and read your textbooks. And make sure you have a thorough understanding and grasp about the resting membrane potential. For those of you who are doing this course with me, we'll also do some simple calculations and questions to help further understand this concept. Until next time, take care.